Okay, I'm going to handle homeostasis, but first let us set up our workspace here. We shall use this template. That one will work. So it's going to be homeostasis and then when you look at homeostasis from your books, you will find out that it has 6 subtopics. These are the main subtopics, and the first one I'm going to look at is the introduction, having general principles. Then the second one is regulation of glucose. Then the third one is regulation of respiratory gases. So I'll call this regulation of respiratory gases. Then the fourth subtopic is temperature regulation. Temperature regulation there. Then the fifth is excretion. And lastly, osmoregulation. So that is what we're going to look at. We've broken down the whole content into 6. The thing is big, but we shall move. So let me try to at least show you what we're going to cover in these subtopics. First, since we are dealing with homeostasis, homeostasis involves self-sustaining systems, or it involves self-regulation, by the way. And in this process of self-regulation, we have to look at organs responsible, and in these organs we have those organs that are going to deal to do the work of detection, so we call them detectors. Then we have controllers, and we have effectors. So the detectors are going to be, as you hear that word detection, these ones are going to be detecting any deviation from a set point. Then controllers are going to be bringing back that deviation to normal. Then effectors are the ones that are the controllers give instructions to effectors, and these effectors are going to be doing the work. They will do the work of bringing back a deviation to normal. Then when you look at this part of the general principles, in our general principles, mainly we shall look at the feedback mechanisms. Feedback mechanisms, where we have the positive feedback and the negative feedback. Then, under regulation of glucose, we know very well that the liver is going to be spearheading this regulation. So the organ involved is the liver, and our detector is going to be the pancreas. Specifically in the islets of Langerhans. Yeah, that's where the detection is going to be, and our liver will do the work of regulating that glucose. So when you move on to regulation of respiratory gases, which gases are these? We are going to look at mainly carbon dioxide and oxygen. And the organs responsible, we have mainly the lungs in our regulation. Then when you move on to temperature regulation, there we shall talk about the skin. We talk about the hypothalamus, and many. So there are very many organs. We shall see them. Then in excretion, some of the wastes we are going to be looking at here. You remember your O-level work, where in excretion we have wastes to deal with, and among them we have nitrogenous wastes, and non-nitrogenous wastes. So when you move on to osmoregulation, as you see, that word osmotic pressure, osmoregulation, osmolarity, it deals with water. So for us, we shall look at water and solute. Let me make it clear a bit. Water and solute balance. So this is what we're going to look at under our topic, homeostasis. So from that, I'm going to move directly to our general principles. I'm going to be a bit fast. So general principles. We have one is our definition of homeostasis. And homeostasis is defined as the mechanisms mechanisms which maintain a constant internal environment. So those ones, when you talk about homeostasis, just we are talking about mechanisms which maintain a constant internal environment. And when we talk about internal environment, the internal environment of someone is that immediate surrounding of a cell. So internal environment is the surrounding of a cell. That's where you find the tissue fluid, and very many others there. So factors that we look into, all factors that should be maintained constant in this topic. Let me try to break them down here. Factors to maintain constant. What are these factors that we need to maintain constant under homeostasis? We need to maintain chemical composition. So the chemical composition of our internal environment should be constant. Chemical composition. So under chemical composition, we have So under chemical composition, I'm having one of the chemicals is glucose. And others are the ions within our body, like sodium ions, the potassium ions, calcium, all those ones. So we need to maintain these ones at a constant. Then, and the other factor to be kept constant is osmotic pressure. Osmotic pressure. And you realize that osmotic pressure is going to be moving hand in hand with the water and the solids in the body. So when we talk of high osmotic pressure, it means that body, or maybe a certain cell having a higher osmotic pressure, has a higher solute potential, or the content of solids within that body or that cell is high. So we move on to the next, which is our part C, which is carbon dioxide. So carbon dioxide level is also a factor to be considered here. And our part D is temperature. So temperature is also among them. So what are the importances of homeostasis? When you're told to state them, importances of homeostasis. One, we have maintenance of pH for the enzymes is one of the importance. So pH maintenance is one of the factor, or one of the importance of this homeostasis. You will find out that if pH is not kept constant, enzyme activity is going to be hindered. Remember, from nutrition, and we look at enzymes as biochemical catalysts, but their activity depends on the pH in which you're going to, or the enzyme is going to be acting in. So if you change this pH, you find out that a certain enzyme is going to get denatured. So enzymes are sensitive to pH. So one of the importance of this homeostasis we are talking about is that it is going to help in maintaining pH for our enzymes. Then the other factor is homeostasis helps in maintaining cell shape. maintains cell shape. Why? Because, for example, if we have an amoeba, or maybe let me say cell A, our cell A. If we put this cell in, maybe if it has a certain degree of solid potential, maybe we say that cell is more concentrated compared to our dilute water in a beaker. If we put this cell in that beaker there, do you know what is going to happen? Water is going to move from regions of its high concentration, because now if this is dilute water, then water is concentrated here. The concentration of water is high. So water is going to be moving from regions of its high concentration to regions of its low concentration. To its low concentration there. So what happens? This cell has to develop some homeostatic mechanisms of maintaining its water content constant. You will find that in osmoregulation. So it has to osmoregulate under homeostasis. How? It has to collect this water and maybe collect it in vesicles or vacuoles and find means of getting rid of that water. So when it gets rid of that water, then we will say that this cell has maintained its cell shape. If it doesn't do that and water enters excessively, when you look at cell physiology, this cell will burst. So if there is no homeostasis, so the cell will end up bursting. Oh, yeah. So it will end up bursting. So when you read about the opposite of this, maybe our cell, this animal cell, is placed in a container having water, but that water is more concentrated than the cell contents. What happens? Water is going to be moving from the cell to the container, or to the region where water is at a lower concentration. So in the end, this cell is going to be losing water, and as it loses water, it's going to shrink. So go back and read your homeostasis. You read your, cell physiology. You will find all that. So when we move on to, let me leave these things here. They will help you in your revision, maybe, if you're reviewing this work. So we move on to the other importance The other importance is this homeostasis makes organisms independent of the external environment. So it makes organisms independent of the external environment. How? I'm going to look at this on the side of temperature regulation, because under homeostasis we have temperature regulation there. When you look at this part, temperature regulation. So from this point here, it makes organisms independent of the external environment, meaning under temperature regulation, organisms like people, so organisms like we people are not limited to our environment, whether a certain environment is in summer or winter. We organisms, more so mammals, by the way, let me say mammals, these ones are not limited in that they can regulate their internal temperatures constant, such that whether the external environment changes or remains constant, it doesn't affect them. Are you saying that? That's why in the North Pole or the South Pole, where we know very well that those regions are too cold, you find that there are some mammals living there, the polar bears. Those organisms are surviving within those regions. Why? This homeostasis is making these organisms independent of the external environment, so they can still survive there. Then we move on to control mechanisms and feedback. So control mechanisms and feedback. So under this we are having positive feedback and negative feedback there. So when you look at this, I will just illustrate it. I'll just have my simple illustration here. Maybe let me carry this. I'll carry that to page 2. So there we are. So control mechanisms and feedback. I have my simple illustration here. We have that circle there, and that circle there, then that one. And within this circle, the first one, we shall call this one the detector. And this one the controller. Lastly, the effector. So, under this, maybe we do this. We have input and output. What am I trying to illustrate? This is the arrangement of all systems we are going to talk about. They have similar arrangement of organs. So, from what I'm having here, the detector can be your skin, the detector can be that pancreas you're having there. It's going to be detecting an increase in sugar levels, glucose levels. So when the detector does so, it sends a message to the controller. So we shall say that our detector, this one, signals the extent of any deviation from our norm. So you'll find many books saying norm. Norm is set point. It's like the normal. So a norm is a set point in biology. So our detectors are going to be signaling the extent of any deviation, and the messages are going to be taken to the controller, and what the controller does, it coordinates our controller is going to coordinate information from many detectors and sends instructions to correct the deviation, and sends instructions to correct the deviation. So that is it. My major interest there is instructions to correct the deviation. Then the effector. What the effector does is, this one is going to bring about the necessary change needed to return to norm. So this one brings about change needed change needed to return to the norm of the set point. So that is it. Then this one is what we call the feedback loop. So after causing that change, or maybe if the sugar levels were high within this region, the detector is going to detect that increase in sugar levels, send it to the controller, then the controller tells the effectors to bring down the sugar levels to the set point. Then an output in form of a message is going to be sent to the detector to stop its activity, such that if our sugar levels were very high, and now it has been brought back to the set point there, we have to give the detector a signal to stop bringing down, or the detector, by the way, it's going to be detecting that change or that restoration of the norm, such that we don't have these cases of continuous deviation down. So that is the feedback loop. That's what we call the feedback loop. Information has to be rendered or has to be delivered to the system to tell it that the deviation that was there has been restored to the set point. So let us look at the negative feedback. Negative so negative feedback there. What's negative feedback in homeostasis? This one is a homeostatic control mechanism where a deviation in the parameter of a tissue or the tissue fluid or blood sets up a series of events which correct and bring about the deviation to the norm. So our definition is going to be, first, we have to identify that it's a homeostatic process. So it's a homeostatic control mechanism where a deviation where a deviation we've said homeostatic control mechanism where a deviation in the parameter of tissue or the tissue fluid or blood sets up series series of events which correct and bring back the deviation to the norm. So what are these parameters? What the parameters we're talking about are those things that we need to maintain constant. These are the parameters we're talking about. So when you look at negative feedback, it's a homeostatic control mechanism. So I'm more interested in control mechanism. Where is it found in homeostasis? Where a deviation in a parameter of tissue fluid or blood sets up series of events which correct and bring back the deviation to the norm. Then we have the other part, which is positive feedback mechanism. So this one is positive feedback. And what is positive feedback? Positive feedback is going to be a homeostatic control mechanism. So we begin with those words: homeostatic control mechanism where a deviation in a parameter of our tissue fluid or blood sets up a series of events to increase the deviation. So the difference between positive and feedback mechanism is here. Series of events to increase the deviation. So in positive feedback, the deviation increases. For example, maybe if your blood sugar level is at 50% after a meal, and your body senses that it's at 50, and instead of lowering it, maybe the body triggers more mechanisms to increase from 50 to 70. That is positive feedback. Instead of lowering, the body is increasing the deviation from a set point. So we have some examples we shall look into. So our examples, these feedback mechanisms, especially this positive feedback mechanism, is a basis of oxytocin release during childbirth. So we see positive feedback in childbirth during. So it's a basis for oxytocin release during childbirth. We know very well that this oxytocin is the hormone responsible for causing labor pain. So it means that in positive feedback, as more oxytocin increases within your body, it's going to cause labor pain in the ladies, and more labor pains are going to stimulate more release of oxytocin, and as oxytocin increases, more labor pains are increasing. Labor pains increase, oxytocin increases. So the thing is like that. Then what I'm forgetting to speak about is examples we find in negative feedback. We have, let me refer them to here. So in negative feedback, the examples are temperature regulation and glucose regulation. So temperature and glucose regulation. So when we move on to glucose regulation, so you find out that at least we've covered one subtopic, our introduction. Our introduction is done. You go through books. I made this summary basing on self-study material, A-level biology. So we shall be going through, it's like I'm going to be reading with you people. This biology is all about reading. You're reading, rereading, like that. It's not like math. Math you can, if you practice a certain concept, there are times when math becomes part of you. But this biology, it needs memorization. This biology needs memorization. So you find out that we people have different categories. You find out that maybe someone reads the biology in one reading, the person has understood everything, and that person is capable of memorizing that content for some good time. Then you find out that there is some guy out there who is going to be, who has to read a certain concept like 3 times today, tomorrow, maybe after a week, to keep this content fresh in our mind. So you have to do the reading. You have to read. You have to sacrifice much of your time reading. Even when you understood the concept, you read. Because concepts have to be fresh in you. Biology, it is strict. It deals with life. So you have to keep it fresh within you. So what I'm going to be doing, I'm going to summarize self-study material. So we shall be using self-study material. That book, it's what I read. It's what I read, and I have an A. I have an A in biology, so I'm going to help you. At least we shall be helping each other in reading. So when I go through such work, go back and read that book in that particular area. For you Form 5s, Form 6, it's like we are revising. Then Form 5, write the notes. At least summarize whatever I'm putting down, you write it. It's like when I go through and you watch this video, your brain is going to personalize this content. It's not going to be like you're reading alone. You're not reading alone, and moreover, you're reading someone's summary. It's difficult to summarize someone's understanding. But if someone goes through their summary with you, then life becomes easy. It's what I'm going to do. So the next recording is going to be general principles here are done. Our general principles are done. So the next recording is going to be for regulation of glucose. So this one is the next.